Welcome to the MPG primer series. I've been asked to come and give you a talk on what is mitochondrial genomics. So I'll give a very basic talk about everything to do with mitochondrial DNA, some with RNA. This is for you guys. You should interrupt me freely all the time. I have a bunch of stuff I'd like to get through, but if we don't get through it all, that's totally fine. So make it interactive. That will make it far more fun. And my name is Sara Calvo. I'm a computational biologist working. Is it not on? All right, but you can't hear it? All right, let me move it closer. How's that? Now do I have a nice low voice? Okay, perfect. Thank you, Erica. My name is Sara Calvo. I'm a computational biologist working in the field of mitochondria for the last 13 years, working in the laboratory of Vamsi Mutha. We study everything mitochondrial to do with basic biology and disease, looking at also biomarkers and therapeutics. But today I'll just talk very basic introduction to mitochondrial genomics. So all of you sitting here in this room right now use about as much power to get through your breakfast as a 100-watt light bulb. Just at rest, not even when you're moving around. So approximately 100 kilocalories per hour. Different organs take up different amounts of that energy. Interestingly enough, the brain, our favorite organ, takes up about 20% of the energy needs of our body, despite that it's only about 2% of the mass. So where does all of this energy come from? As you know, there's no organ of energy production. It's not like we have a nuclear factory sitting somewhere in our body. It's a distributed system. The energy is produced where it's needed, locally in every cell, and the site of this is sitting on the inner membrane of the mitochondria, this beautiful structure that's within our cells. And in particular, this structure has a double membrane, and the inner membrane is incredibly convoluted and packed with the machinery to do this process called oxidative phosphorylation, which is a mouthful, but this, I want to convince you, is the most amazing evolutionary pathway there is on the planet. This creates energy. Like, how do you create energy from nothing with no pollution? This is our system to generate ATP, and it was discovered in the 60s, and I'm going to walk you through this amazing pathway, which many of you may have learned in biochemistry, but maybe not with the same enthusiasm that I hold for this pathway. So, it all is based on a membrane. So you have a membrane, and you create a gradient across this membrane. And then, so that's complexes. There are 4 complexes to create a gradient across the membrane. And then there's a complex, complex 5, which uses this gradient. It's an electrochemical gradient, the gradient of hydrogen ions and electricity. And then it uses this gradient like a water wheel. And every time a proton goes through this channel, there's a rotating motor, and it sticks a phosphate on an ADP molecule, making a high-energy phosphate bond, creating this molecule ATP. This is the energy currency of our cell. It's a tiny molecule. You may know this molecule better for the A as an ATGC in the genome. It's the same molecule which is used as the main energy currency in the cell. That last bond, the phosphate in the ATP, triple phosphate, has a lot of energy, and to power most of metabolism and other working processes in the cell, that you pop off that phosphate, and that provides energy to do the work in your cell, in the cell and in organisms. So that is, you create this gradient, and then you use this ATP synthase to turn ADP into ATP. So that sounds great. You have a gradient. It's like a water wheel, churning out ATPs over and over again. So how do you create a gradient without using more energy than you generate? And that is the amazing power of oxidative phosphorylation. It starts with a high-energy electron. So think of your model of the atom. Very, very high-energy electron that's sitting on a molecule called NADH. That high-energy electron gets passed down a chain to lower and lower and lower and lower energy states. And at the end of the day, it's grabbed by an oxygen. And that pull of the oxygen that really wants that high-energy electron, every time it's passed down this electron transport chain, it has a little bit of extra energy that's used as it's going down. It can use that to translocate protons from the inside of the mitochondria to this intermembrane space in the mitochondria. And how it's coupled is pretty amazing. It's not like there's a channel that opens that lets the hydrogen go by. It's not actually known, despite all the years of study, how this works, but you change the conformation of this huge complex 1, 3, or 4, and you get a little column of water, and a hydrogen ends up on the bottom, and a different hydrogen pops off the top, and you've translocated a hydrogen that's powered by this electron going from a high energy state to a slightly lower state. So in complex 1, pumps hydrogens across, passes the electron to the next one, to the nenext complex, that pumps protons across, passes the electron over to complex 4. Oxygen really wants that electron, takes that, and it converts oxygen to water. And at the end of this electron transport chain, you have a high gradient of hydrogen on one side, and then that is what powers this ATP synthase. So this is conserved throughout all life. This is maybe a simplified version of this was probably in the first organism life on this planet. We have a more complex one now, but this is the fundamental system. There are other ways of making energy, but this creates 90% of the cellular ATP that we use in our body is created from this. And as you see, one of the inputs here is oxygen. This pathway is the main reason we need to breathe oxygen in the world. So when the oxygen we extract from the air, 90% of it goes to this pathway to create our energy, our ATP. Now the pool of ATP is, if you added it all up, it would be about the size of a teacup. But it's continually being recycled, ADP to ATP, and it's being used at an incredibly high turnover rate. And this system is continually turning over the ADP to the ATP. If you had a deficiency in the system and you wanted to eat the ATP that you needed, you'd need to eat your weight in ATP every day. You could order it from Sigma, several $1 million a day, could supply your energy needs, but it may not get where it's going. And all of this requires the membrane. The membrane is the key to this energy generation system. And so this membrane is folded up within the mitochondria, which is within the cell. If you took all of them out and laid them out, people calculated it would be about the size of a soccer field, is the amount of mitochondrial membrane surface area in your body. So you have a huge amount of, but it's all packed in really, really tight so that you can do this process all the time. You can sit here, eat, think, blink, all. All right, so I wanted to give you a motivation why you care about mitochondria. This is why I am in love with this organelle. Any questions now? Get into the genomic side. Okay, so for this talk, I'll tell you background about mitochondria in general, its evolution and diversity, some of the challenges for analyzing the mitochondrial DNA sequence, because it's slightly different from the autosomes that we're all much more familiar with here at MPG. Then I'll get into more disease-related part of what does rare mitochondrial disease look like and how we diagnose it in the clinic. And if we have enough time, there's some recent work that I like on somatic mitochondrial DNA and what that teaches us about evolution and mitochondrial changes. So again, interrupt me. No question is a dumb question. For those of you who came in, my name is Sara Calvo. So I can just say, hey, Sara. All right, mitochondria. The name comes from the Greek mitos, meaning thread, and chondrion, which is granule. And these were discovered back in the 1800s when microscopes were developed. You could get more and more resolution within a cell. They were first seen in the 1840s, called bioblasts, and then Carl Benda in 1898 renamed them mitochondria, which is the name that we still know and love them by. We're used to kind of seeing them in this kidney bean shape. The more accurate way to think of mitochondria is a network, much more like the ER. It's this network that's continually undergoing fission and fusion. It's moving around within the cell. And so you can see these beautiful live imaging fluorescence to see these guys zipping around the nucleus. So here's the nucleus, and this is this mitochondrial network that's surrounding the cell. And here's a high EM of that. Now, you may be familiar with EM images. So different organs all have mitochondria, but the mitochondria do slightly different things depending on the energy needs or depending on other biosynthetic needs. And depending on the tissue needs, the mitochondria have different morphology and structure. Here are just some of the different ones that look quite different from one to the next. They all have a double membrane, though, because you need that membrane to do OXPHOS. Some recent work has shown that the different mitochondria, they can fuse and fizz, but they also can form synapses within the cell, and those can be electrically charged. So the whole way the OXPHOS system works is you create this energy, this electrochemical gradient within one mitochondria, but those can be have like junctions between them, and this may be responsible for why you can get things like the muscle physiology to work, where the oxygen is coming in one place, but you actually need the ATP several millimeters away. So you can have these like relay stations in your power grid. This is just showing some more junctions of these between mitochondria and different organelles. And of course, mitochondria do not exist by themselves floating. They are part of all of the other stuff that is going in the cell. Here are these double membrane structures, but they're intimately interconnected with the ER, with the nucleus, and other structures, the peroxisomes in the cell. There are many different cell types have different energy needs. They have different looking mitochondria, and they have different number of mitochondria. The cell with the most mitochondria is also the largest cell. The human egg will have hundreds of thousands of mitochondria just waiting there. And then once it gets fertilized, you start doing divisions. You're not making more mitochondria. You're just apportioning those 100,000 mitochondria into different cells. Energy needing tissues like the heart, skeletal muscle have super high amount of mitochondria. Things like blood have very low amounts of mitochondria. And as you all know, if you're talking about mitochondrial genomics, mitochondria have their own DNA. This is a remnant of a bacteria that at the earliest time in eukaryotic evolution was engulfed by the proto-eukaryotic cell. And it remains, the mitochondria remains very much like a bacteria, and it has a genome like a bacteria. It's circular. Here's an EM of a mitochondrial DNA. And here's one replicating, so now it has two loops. It hasn't yet broken apart. And this was discovered in the 60s. So it has this tiny circular genome. Now this genome is 16 kb. This, I can't emphasize enough how tiny this is. A typical gene in the human genome is about 75,000 bases. This is 16,000 for the mitochondrial DNA. And it is chock full of stuff. It has 37 genes, only 13 protein coding genes, but some tRNA and ribosomal RNA that help make these. And unlike the nuclear genome's genes, pretty much every single base is under selection and doing something. There's practically no intergenic space. It's gene, 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 gene. Here you can see the protein coding genes are in like dark orange here, and in between them are the tiny tRNAs, which are about 75 base pairs. So it's like protein coding gene, tRNAs. Every base is useful here. It has a slightly different genetic code from the standard one, so if you're doing genomics analysis, make sure that it's using the right genetic code, otherwise you'll end up with weird stop codons that aren't really there. And it has a mutation rate that's about 10 times higher than the nuclear DNA. But it's under extremely high purifying selection. The one part of the genome that's under not so high purifying selection is this control region called the D loop. It's called the D loop because when it's replicating, it has a third little bubble here, and it actually looks like a D under the microscope. And this is hypervariable among people. It's not just neutrally evolving, like the third codon positions, the synonymous positions. This is hypervariable among people. We have no idea why it's hypervariable. One of the projects I'm doing is trying to answer the question why it's so variable among people, but because of its history of being variable, it's been used as a marker of ancestry, maternal ancestry, for decades now. And if you do a 23andMe, you'll get your maternal ancestry from the mitochondria, but mostly just from like 200 bases here in the D loop can tell you about your maternal ancestries. In terms of the transcription, that's the DNA loop. RNA, it's polycistronic, also like a bacteria. You start in one place and you transcribe the whole heavy strand, and on the other side you start at the light strand promoter and you transcribe this. And then it's polycistronic. Those genes have to get cut up and then translated to make protein. So it's a totally different system from the nuclear system. No introns, and it's under so much selection that in 2 cases you actually have genes that overlap in different frames. And that is highly unusual in genomics. That's cool. Any questions there? I'm super clear. Good, yes? Almost all the genes are on what's called the heavy strand. It's the heavy strand because it has more Gs, so it literally is heavier if you run it on a band, a gel. And then there's only one protein coding gene on the other strand. Yep. As you guys know, this genome is maternally inherited. That's for a couple reasons. One reason is the egg has like hundreds of thousands of mitochondrial genomes, and the sperm has very few. Most of the sperm mitochondria are sitting in the tail that never even make it into the egg. But the few mitochondria that are in the head of the sperm that make it into the egg are tagged for degradation specifically. So that's why there's incredibly low paternal transmission, and that it's targeted, it's ubiquitinated, and then ground up by the proteasome. So if you block that, you actually get more paternal transmission. There have been a bunch of papers to try to look through for paternal transmission of mitochondria in different species. If you look hard enough, you can find very, very few examples of this, looking at mouse back crosses. There's a single human case in 2002 showing paternally derived allele. And then just a couple months ago in PNAS, they showed several different families that had paternal inheritance. Again, this is incredibly rare, and that might tell something about the biology of what's going on in those families. Another concept in mitochondrial genomics that's totally different from autosome genomics is because you have many copies of the mitochondria, if you have some deleterious mutation or any variant, it can exist at any level of heteroplasmy in the cell, which means like what percent of the molecules have that variant. It could be 70%, it could be 2%, that is called heteroplasmy, percent of all the molecules with some mutation. Now, if you can imagine if you have a really bad mutation arising, if it's at 1 or 2% of all of your mitochondria in that cell, it's not going to have too much of an effect. So you need to get to a certain level of heteroplasmy before you see a phenotype in that cell or in an organism, and that's called the threshold effect. And it depends on the exact mutation, but often you need about 80 or 90% of your mitochondrial genome molecules to carry the mutation before you'll see a phenotype. Make sense? Heteroplasmy? Okay. Because of this phenomenon of heteroplasmy, you end up with different organs can have very different levels of a deleterious mutation. And that often can happen right at the beginning of life. You go through a bottleneck. So that egg has hundreds of thousands of mitochondria. Some of them carry the deleterious mutation. It can turn out that certain organ systems will carry a higher mutant load than others. So you could end up, what we often see in our patients is there'll be a deleterious mutation that you see in the muscle, and it causes a muscle myopathy. But if you look in the blood, you don't see it at all, or it's at a very low heteroplasmy. And that can become because of this bottleneck that you go through in evolution early on in the egg development. I love this study showing that this may not be a random process of how much mutant load can be tolerated in different organs. So this is a paper from the JAMA Neurology that looked at monozygotic twins that have a really bad mitochondrial DNA mutation. This one here in a gene called ND6, which is the one gene that's on the light strand. So the mother has this variant, but it's at low heteroplasmy, and she's completely fine, no symptoms. The monozygotic twins, the one that came in first, the proband, had this bad mutation at 84% of all of the mitochondria in the skeletal muscle, and in fact they have they have optic atrophy, ptosis, which is like droopy eyelid, and epilepsy. So the muscle had really high levels, hair follicles had high, buccal mucosa, high, blood had very low. But what's interesting is the two twins show extremely similar levels of the heteroplasmy. So I think there's some difference in selection that's happening in the different organs that might allow a deleterious mutation to take effect in the muscle, but not in some of these rapidly proliferating tissues like blood. The other corollary from studies like this is if we're interested in diagnosing diseases of the mitochondrial genome, typically what we use for genetics is blood samples. For mitochondrial disease, blood is the actual worst tissue to look in. There could be really high levels of heteroplasmy in the affected tissues like the brain and the muscle, but the blood may be at like 1, 2, 3% heteroplasmy. And again, these rapidly proliferating tissues tend to lose out the mutations. Another issue with the mitochondrial genome that ends up being a pain in the butt for an analysis is an issue called numites, nuclear sequences of mitochondrial origin. So what this means is that the mitochondria here in purple, the mtDNA in purple, occasionally escapes out of the mitochondria and gets integrated into the autosomes. And once it's sitting in the autosome, it's just sitting there, it picks up mutations, it travels along, it gets inherited. It doesn't really cause much trouble for any of us. This has happened across evolution. We see it in all sorts of different species. We see it in humans. But for analysis, this is a pain in the butt because it causes alignment problems when you're doing sequence and alignment. So if you take the mitochondrial DNA and you blot it against all the autosomes, you just look for similar sequences. There are about 750 of these numites littered around the autosome. Some of them, most of them are short and moderately diverged, which aren't going to cause big problems, but others of them are long, like 5,000 or 10,000 letters long, that can have areas of incredible similarity. So this causes mapping issues when we try to do sequence analysis. The insertion is not random into the genome. It goes typically into open chromatin like you might expect, like other things. And this integration is still happening. So if we looked in this room, some people will have more of these numites than others that are not in the human genome assembly, but they're just integrations in some people and not in others. All right. Any questions there? Great. I will move on to mitochondrial evolution and diversity. So I love evolution. This is one of my favorite topics. So in the 3 domains of life, how many here learned about the 5 kingdoms of life in school? How many learned about the 3 domains? All right, so the 3 domains is the way more modern approach. But if you were educated long enough ago, you heard about, animals, fungi, plants. Throw that out. The tree of life really has 3 main branches. They're bacteria, single-cell organisms, very small, only a couple shapes. Archaea, also single cell, also really small, more shapes. And then everything else, which is eukaryotes. Eukaryotic cell is enormous compared to either the bacteria or the archaea. These arose first. There's fascinating history of, or theories of the evolution of life. And some theories, one of my personal favorite is life evolving in the deep sea vents, in alkaline vents near the ocean floor. And that bacteria, what became bacteria and archaea evolved actually at the same time, way back in early evolution, and then had these separate trajectories until this one event that's only happened once in the history of the planet of the beginning of eukaryotes. And eukaryotes happened around 2 billion years ago. Most of the nuclear DNA came from an archaeal host, but some bacteria. And there was an endosymbiotic event of a bacteria getting trapped within like a cell, and that was the ancestor to our mitochondria. And it happened right at the root of eukaryotic biology and probably is part of the selective advantage of why you have eukaryotic life, which can be, the cells can be very big, partly because you have energy generation within the cell with this convoluted inner membrane. You can't have that in a bacteria. Okay, so this endosymbiotic bacteria was an alpha-proteobacteria. That's the kind of bacteria it was. And it evolved into our modern-day mitochondria, which you find in practically every single eukaryote that you look at. There's one example of a species that has fully lost its mitochondria called monocercomoides, and we now know how it gets along without having a mitochondria. But every other organism that we've ever found of eukaryotic have a mitochondria. They have this double membrane I told you about, mitochondrial DNA, and that's just one part of the eukaryotic cell. You have other things in eukaryotes like a nucleus, the ER, all these other structures that other people care about. And then there was a later endosymbiotic event of a plastid that became chloroplasts and the apicoplast and things like that. This proteobacteria ancestor was a bacteria. It had like 5,000 genes. The modern day mitochondria only has 13 protein coding genes in humans. All those other 1,000 genes got transferred to the nucleus or got lost and replaced by their archaeal homologs. Now, mitochondria are quite different across these 2 billion years of life, as you can imagine. Most things have changed a lot in 2 billion years. The size of the genome is radically different. So ours is 16 kb. here, Homo sapiens. There are ones that are even smaller. Plasmodium, the malaria parasite, has only 3 genes that are left in the mitochondrial genome. The other ones have all managed to escape to the nucleus. Arabidopsis has a genome that's like way, way, way bigger, but it has a lot of non-coding regions. So ours is maintained this beautiful structure where every base matters. Others have fluff, either introns or non-intercoding intergenic regions. The number of genes also is not quite as radically different across species, but still varies from 3 to like 90, and Reclidomonas has the most number of genes. And we think of the architecture of the human mtDNA, a loop like bacteria. Other organisms have gone other ways. There are organisms that are linear. There are ones that have a gazillion tiny little chromosomes with telomeres to maintain it. And there are ones that have like a major loop and lots of little ones. So the field of mitochondrial diversity is pretty cool. And some of these genomes actually have editing. Like there's so much RNA editing that you can't even tell where the genes are originally because they're so heavily edited. It's pretty amazing what evolution can do if you give it enough time. Other organisms, mitochondria, have introns, telomeres, editing, as I just said. So, all right, so one of the things I've been working on this last year is teaming up with DSP here at Broad to come up with a good pipeline so that every one of our whole genome sequences that are being done here at Broad, when you get your VCF, it will have not only your nuclear variants, but also really high quality mtDNA variants. But there are a couple analysis challenges that make it different from the autosomes, so you have to deal with it differently. It's circular, so that at the ends, if you do, what we typically do is you cut the circle and then you just align it, and then you have problem at the quote ends, just because we don't have a circular aligner that's very good. Because there's so many copies per cell, you need to call variants not just heterozygous, homozygous, you have to call it at all levels of heteroplasmy. These new mites end up being a huge pain in the butt because the reads are going to the nuclear genome rather than to the mitochondrial genome, or vice versa. Things that should be autosomes are going to the mitochondria and causing false positives. You have to annotate your things properly, and there's a high mutation rate that changes some of the assumptions of our callers. One of the good sides is that we have excellent coverage, which means that you can tackle some of these problems. So a whole genome done here is typically about 30x for the nuclear genome. It's about 3,000x for the mitochondrial genome, so that means we can actually call low heteroplasmy variants quite well. I'm also looking at RNA-seq. Blood has the lowest level of RNA, but you still get about 3,000x coverage of the mitochondria. Muscle, you end up with 100,000x coverage or 200,000x coverage of every base of the mitochondria in the RNA-seq, and that presents its own analytic challenges dealing with that level of coverage. Here's one example of a new mite problem. So most new mites aren't a problem, but there are 2 that are like the main problems. And here's an example of one of them on chromosome 1. So here's the mitochondrial genome here from position 1 to 1600, and this is the coverage. So it's pretty even coverage, except you see these huge dips here. Do you guys see that? So this is one African sample. And then if you look at this particular region on chromosome 1, that's a new mite. It was originally came from a mitochondria thousands of years ago. The reads are all going over there. So this should have 30x coverage, and then up you come here, it has 1500x coverage, because those reads should have come from here. So that's a mapping problem. So that's one kind of mapping problem. Oh, maybe I didn't show a different one. But you end up with these huge stacks of mapping quality 0 reads where the aligner doesn't know, does it come from the mitochondria? Does it come from chromosome 1? We'll throw up our hands and just stick it somewhere randomly. And so you end up with this, and then you can't call variants with a mapping quality 0 region because the algorithm doesn't know if the variants really come from there. So you need a separate pipeline for dealing with this. It's technical, but if you don't get these right, you end up with a lot of false positives and false negatives in your data. No one wants that. But working with DSP for the last 6 months, we now have a pipeline in FireCloud. We're still working out some of the bugs, but yep.
Is that alignment issue something that would be avoided if you're only capturing exome? Because they don't.
With the exome, if you're doing the exome?
Because they're only in intronic or intergenic.
Yep, so the exomes have 2 problems. So the old exomes from Agilent cover the mitochondrial DNA just fine at about like 70x coverage. And then there are regions in the nuclear, you're not supposed to be getting the mitochondrial DNA, but there's just enough off-target reads that you end up with about 70x coverage of the mitochondrial DNA. And then random places in the nuclear DNA, you'll end up with the stack of reads that are supposed to come from the mitochondria, but are now going to the middle of chromosome 1. So you probably wouldn't see them because they're not in your bait set. But if you're trying to analyze data from exome data, you still end up with the new mite problem, the alignment problem, just because of the mapping, not because of the capture. The more recent exomes, like not SureSelect, the Illumina exome, got really good at getting rid of all of the mitochondria reads. So they put in blockers to the mitochondria, so you ended up with no coverage for the exome. If you put baits for your mitochondria, then you end up with ridiculous coverage. So DSP, I've been advising DSP, and we now have a pipeline that's pretty great, but we're still working out the last bugs. Again, the tools have not been developed for this, so we have to deal with all of these, like the high coverage, the looking for low heteroplasmy. So we're getting there, but I think in the next month or two, we'll have a pretty awesome caller. So every one of you that gets a VCF from a whole genome sequence will now have mitochondrial variants sitting in there, which is great. We've run this already on nomad whole genomes, 17,000 nomad whole genomes with Kristen, who's here. She's run this on 17,000 samples, and so we can build a database of population frequencies of mitochondria, both the high heteroplasmy variants and the low heteroplasmy variants. And so for clinicians, this will be super useful. The same database that you're going to look at all your other allele frequencies will have the mitochondria. I've also been running it on RNA-seq samples. This presents other challenges, but there's also some huge gold here, and I love this analysis a grad student in our lab did. So RNA-seq, you take sequence from, say, muscle, and you sequence the RNA, and the things that are really highly abundant, you just sequence over and over again. And that's a problem for your RNA-seq because you can't find the sequences of the low abundance genes. So if you look, this is a graph for each tissue. This is from GTEX, from about 50 tissues in GTEX. What percentage of all of the reads in your RNA-seq experiment are coming from the top 25 most highly expressed genes? So over here in brain, 70% of all the reads in your RNA-seq are coming from the top 25 genes. And in red are which of those are coming from the mitochondrial genome. And you can see in the brain, about 70% of all of your reads you're sequencing these 13 mitochondrial RNAs over and over and over and over and over again. And so you're not going to be getting to the other, brain researchers are interested in any other things besides the mitochondria that they're not going to see. But if you are interested in mitochondria, this is a gold mine, and because, so this sample will have like 200,000x coverage of the mitochondria, you can do all sorts of things with this gold mine of data. And I'm working on a project with GTEX to do GWAS of mitochondrial traits to say what nuclear regions control these weird traits within the mitochondria that we can measure extraordinarily well because of the depth here. And other tissues like whole blood, it's not very red here, so blood doesn't have a lot of mitochondria, but here you can't read it, but hemoglobin A2, hemoglobin B, those are the genes that in blood you're just sequencing over and over and over and over again. And so this is one of the joy and problems of RNA-seq is the abundant things suck up all of your signal. That was just a little aside because I find this data so fascinating. Yes?
Can you comment on how many nuclear genes are responsible or contribute to mitochondrial transcription?
Great question. In fact, I did my thesis on this question. I love this question. So there are only 13 protein coding genes in the mitochondrial genome itself. All the other things that make up the mitochondria have genes that are sitting in the nucleus. They get transcribed, ribosomes come, make proteins, and then those proteins get imported into the mitochondria. So there are about 1,200 mitochondrial proteins that are nuclear encoded that go and make up the mitochondria. Some of those are metabolic proteins, but about 600 of them are involved in transcribing and translating the 13 damn proteins that are still in the mitochondria. Like 500. 500
of the 1,200.
Yeah. So things that you have to replicate your RNA, you have to transcribe it, you need your ribosome, you need all of these things. So, other questions? Nope. Okay. So let's switch over to disease. And in our lab, we've been mostly focused on rare diseases to date. So I'll talk about some of the rare mitochondrial diseases. And not just mitochondrial diseases in general, but specifically diseases of OXPHOS. The pathway that I told you about in the beginning is responsible for making about 90% of our cellular ATP, the main powerhouse of the cell. So you could imagine the system is super important. If you have a problem in it, you're going to get various diseases, and that in fact is the case. Each of these diseases is individually incredibly rare, but together they have a prevalence of about 1 in 4,000 individuals. But it's not one disease, it's an umbrella term of many different diseases, and it's very, very heterogeneous in terms of which organs are affected and the degree of pathology in each of these organs. But typically, a mitochondrial patient would have CNS features such as stroke, ataxia, epilepsy, migraines. Many of them have eye manifestations. There's optic atrophy, retinopathy, ophthalmoplegia. In terms of ears, there's deafness, sensorineural deafness is one of the main features. There's cardiomyopathy, myopathy, muscle problems, liver failure, anemia. One physician said basically any organ can be affected in mitochondrial disease at any age of onset at any time. So these are very, very heterogeneous. If you're a clinician, these are a huge problem because the symptoms are often nonspecific, and separating out things that are really caused by OXPHOS disease versus many other pathologies can be a problem. So diagnosis is hard. It's recently become a diagnosis of last resort. You don't know what it is. You say, oh, possible mitochondrial disease. And so that's also a problem. And even worse for our patients is even if you get a really clear diagnosis, there is no proven therapies. So for our patient population, it's grim times, but there are now, I think, 7 clinical trials. We have some interesting therapies on the horizon, but currently nothing. In the past, the way of diagnosing these diseases was using these like Chinese menu carts. This is the Morava mitochondrial disease criteria in children. That still is used, where you get points. So if your child patient who's coming in has ophthalmoplegia, you'll get a point. If you have muscle weakness, you'll get a point. So you maximum 2 points from the muscle symptoms, 2 points from the CNS symptoms. You add these all up, imaging, some of the morphology. You add them up, you get a score. And if you have a score 2 to 4, you have possible mitochondrial disease. Score 5 to 7, probable mitochondrial disease. Score 8 to 12, you have definite mitochondrial disease. Now, when I was a graduate student, I thought this was ridiculous. I'm like, I'm going to clean up this field. Genomics, it's going to be amazing. I had my thesis on what are all the nuclear genes that go to the mitochondria. These are going to be the causes of rare Mendelian mitochondrial disease. If it's not in the mitochondrial DNA, it's in the nuclear DNA that creates proteins that go to the mitochondria. And we can replace all of this, single blood test, do the analysis, we're going to solve this. This is going to be ancient history. Now, like many things in biology and medicine, it's never that easy. And I think I'm continually to be awed by the complexity of how biology and medicine work. So we have made great progress, our group and others, in really helping these patients, but there's a really long way to go. I got out of let me go back to this. So I wanted to give you some sense of on the clinical spectrum, what the heterogeneity of these diseases under the umbrella term mitochondrial diseases. So on one end of the spectrum are the most severe cases. These are often babies who will be diagnosed within the first days or up to year of life. The expectancy is under 2 years, although there's some kids who can make it to childhood. So it's called Lee disease, and it's diagnosed based on very specific MRI findings. If you have this MRI finding, you have Lee disease. It is caused by mitochondria. This is kind of pathognomonic. So that's the very clear case and the most severe case, Lee disease. There are over 75 different nuclear genes that can cause Lee disease. So it's very genetically heterogeneous, but clinically we have a beautiful biomarker that makes diagnosis of the severe form quite good. Another mitochondrial disease I would put at the other spectrum of the least severe is LON, Leber's hereditary optic neuropathy. This is often seen in young men like your age, sitting here, perfectly healthy, and then in their 20s, they first go blind in one eye, painlessly, and then a few months later go blind in the other eye. And that's their only symptom. They also have a mitochondrial disease. It's caused by a particular lesion in the mitochondrial DNA, specifically in complex 1. This mitochondrial mutation is present in every cell in their body, and yet their only symptom is blindness. It's amazing. And we don't know why you get this very specific pathology for this mutation. But that's just like, these two diseases look completely different. Kids dying in a couple days versus men going blind at age 20. They're both mitochondrial diseases. Genetically, they're incredibly, the umbrella term is genetically heterogeneous. There are over 145 genes. This isn't like a GWAS, 145 genes. These are 145 different genes that each cause monogenic disease. So highly penetrant inherited diseases. Some of them are in the mitochondrial DNA. There are 13 proteins that accounts for about 15 to 20% of pediatric patients, more of the older patients. And these are maternally inherited. But most, particularly for pediatric cases, are caused by mutations in the nuclear DNA. In the 1,200 proteins, that's most of the cases. And these can be inherited recessive, dominant, X-linked, you name it. So what makes these problematic to diagnose? Diverse symptoms, often nonspecific, many, many genes. And for the most case, you can't predict which gene is going to be the cause for which of these symptoms. So that's why I was thinking I can clean this up, do a single blood test, solve the case. And the answer is, is the glass half full or half empty? It is now true that instead of spending 10 years on your diagnostic journey to get a diagnosis, now in about 50% of cases, a single tube of blood and an exome study can give a definitive disease, particularly in the kids. The pediatric cases, a little less good for the more fuzzy suspected mitochondrial disease. So this is amazing. Like, this really is a change in medical practice. This is now the first-line diagnosis for these patients rather than after your years and years of seeing many doctors. So this is great, but have we gotten anywhere near 100%? Not even close. So, question?
Just in terms of the terminology, you say mitochondrial disease,
Yeah.
that you're including in that nuclear genes.
Nuclear genes, so it's OXPHOS disease. Right. So it's, yeah, totally. Okay. And this, I think, is very similar to many other diseases that get talked here at MedPop. Pediatric monogenic diseases, exome sequencing can solve half, maybe 75% of cases, but there's still this big gray area where we can't provide diagnosis. So the good news is we do have answers for patients. We end up with new biology. We find new genes in these pathways, new phenotypes so we can expand the phenotypes for any given disease gene. And then we have this big gray area where either it's not really a mitochondrial disease to start with, or there are variants in non-coding regions, structural variants, that we can't yet interpret. And I think the field is really moving fast, but it continues to be hard as we try to get to the tail of these. And I think the main challenge ahead, again, for many diseases, but mitochondrial diseases included, is interpreting these pathogenic coding and non-coding invariants. When you find a variant, how do you know what the effect is going to be? And there's nothing specific about mitochondria here. Mitochondria has special tools that we can have cellular models so we can prove pathogenicity in a dish that you can't do for things like diabetes. So that's a fantastic advantage, but it's also a challenge of the genetic and phenotypic heterogeneity. All right, any questions there? All right, we still have a little time, so maybe I'll tell you about some recent work into somatic mutations in the mitochondrial disease. Yep. No, don't apologize. This is for you guys.
Specifically, not so much the mitochondrial genes that are in the nuclear, but the mitochondrial genes themselves.
Yep.
Do those have clear, phenotype, genotype associations like the nuclear ones do in the more general case?
Yes and no. So in some cases, There's a particular common, or not common, but variant, like the Milas variant, causes a syndrome called mitochondrial encephalopathy, lactic acidosis, and seizures. So that's a syndrome that is caused by a particular, there are actually 9 different mutations, but one common one. And so most people who have that will eventually get seizures in their 40s, typically. So you do have some genotypic, phenotypic correlation, but you also have low penetrance. Like not everyone with that mutation will get the disease. And then also you look in a single family where they all have the same mutation, and some get this symptom and some get these symptoms. So you also have pleiotropy. So all of the above is the answer. Yeah.
Can you comment on the mitochondrial replacement therapy? Since there was about 2 or 3 years ago, the IOM recommended there should be more research on mitochondrial research.
Yeah, so this has, so for parents where the mom has some awful mitochondria, is carrying some awful mitochondrial disease, until recently there have been limited interventions. And I go to a conference where we see patients with mitochondrial disease, and you'll see these families where like 4 kids all have some incredibly difficult mitochondrial disease. So the community is really looking for therapies. So one, for if the mutation is in the mitochondrial genome itself, there's a controversial procedure called mitochondrial replacement therapy, where basically you take the cytoplasm from one donor, and then you take the nucleus from the genetic mother and father. So these are called 3-parent babies. And there's been ethical investigations onto whether this is safe. In England, the first kid who was created by this procedure is, I think, now 14. She's doing fine. It has been approved in England. The US, there's been a lot of recommendations for doing more study. My boss, Vamsi, was on that committee to try to look into both the ethical and the scientific implications of doing that. Interestingly, it was not known, but there are older research about these fertility clinics that actually had been doing this procedure not for mitochondrial DNA, but just to improve their efficiency and yield of including cytoplasm from like another cell. And there are actually plenty of babies walking around with another donor for part of the cytoplasm. But that had never been, no one really, it even now isn't highly publicized that this has happened. So I think there is more work into it. I personally don't see a huge problem with doing that. There are some theoretical problems, but I think it seems fairly, my personal opinion is that it's safe, and I don't see a problem going forward. It's not nearly as controversial as like a CRISPR baby or something like that. Other questions? Yeah.
Before you get to the somatic mutations,
yes.
Age-related loss of mitochondria,
yes.
Is the process well worked out?
Okay, so this, there's a long literature into mitochondria decline and aging. And this has been seen over and over again. So the quantity of mitochondria decreases as you age, and in some cases the quantity, so both the quantity and the quality, there are higher mutations in older individuals. And as you know, as we age, we all decline in a nonlinear fashion. And so there was a hypothesis that a lot of the causal reason of why we get the phenotypes of aging had to do with mitochondria. So we know that mitochondria do decline. Is it a causal role or is it a consequence? The jury is still out. There's a lot of research into this field. I personally am not very convinced by the causal role of mitochondrial decline and aging. There definitely is an association, but I think the evidence that it's causal is pretty low in most cases. Other questions? No. All right, we only have like 3 more minutes. I'll give you a taste and not go through the whole field, but of a couple recent papers in the somatic alterations in mitochondria DNA. And I'm going to present at a very high level 3 papers that came out a couple years ago. And one reason I love these papers is from the biology, but the other reason is it's a cautionary tale to computational biologists like me of when you look at your data and drawing conclusions that like sound great at the time, but if they're underlying processes that if you don't account for, you come up with a complete wrong interpretation. And so I think if I had done these papers, I would have come to the same wrong conclusion as some of the authors here. And so I love the one paper that showed the more complicated version. Okay, so there's inherited mutations in mitochondria, and they're also somatic. Every cell can pick up more mutations. That can be lead to cancer. It can just be neutral. It can cause decline and aging. And there are several studies, these are just the 3 first ones, that looked at somatic mutations in mitochondrial DNA. And 2 of them were in cancer. Is this working now? Yeah. So this paper actually coming out partly from MPG, Spectrum of Somatic Mitochondrial Mutations in 5 Cancers. This one not looking at cancer, but just at different tissues among 12 people, shows positive selection for somatic mutations. And then this lovely paper looked at somatic mutation across like several thousand different cancers by comparing tumor to normal. So the first paper looked at 200 tumor normal pairs using off-target mitochondrial reads from whole exome sequencing using the older technology when you could do this. And they showed that somatic mutations had a 6-fold enrichment for non-synonymous versus synonymous mutations. And so that is typically what we use as a signature for positive selection. So they concluded that there's positive selection for somatic mutations in these cancers in mitochondrial DNA. This non-cancer paper looked at mitochondria from 150 individuals and many tissues. They also show this strong enrichment of non-synonymous versus synonymous mutations, saying positive selection. So that all makes sense, except that it's just not true. And this third paper did a beautiful analysis, one of my favorite papers, looked at 600 tumor normal pairs, looked at 2,000 mutations, also saw exactly the same as these papers, a 5-fold enrichment of non-synonymous versus non-synonymous mutations. So it looks like positive selection, but they went on to do an analysis of the mutational signature, and there are certain biases in the way, the process of causing the mutation, and once you account for that process, that signature completely goes away. And so I don't have time to go into the beautiful paper to show you how this is, but for me, this is a cautionary tale of over-interpreting the signals that we're getting from the mutation. So I think that was probably a little unsatisfying, but we're running out of time here. So is there anything? Nope, that's all I have for today. So thank you for your attention.